Hi everyone. My name is Richard Suendra. This is going to be a presentation for Physics 430, which is nonlinear dynamics and chaos, taught this semester by Isvan Kovacs. I'm going to be presenting on the topic of biological morphogenesis. If you don't know what morphogenesis means, it means the generation of form. In biology, it refers to the generation of form in organism or a cell or a tissue or anything like that. The term was coined by Alan Turing, which we'll talk about in a bit later, but for now, what are some examples of biological morphogenesis? Well, the first and probably most well-studied one is in terms of animal patterns. So like the stripes of a zebrafish, or how sort of the patterns on Drosophila develop. That's really commonly studied nowadays. Some other ones, developments of organs such as the lungs, kidneys, organelles, things like corals or cancerous tumors and things like that would be included, and just a lot more. So throughout this presentation, I'm gonna have these little boxes, those white boxes, as you see on the top left of the slide, with a purple number in the middle, and that's gonna be the paper that I'm referencing at the end. So at the end, I'm going to have a list of all the references I have, and the number will correspond to the paper that's on that list. So before we start talking about some of the other newer things, let's bring it back a little bit with a little history lesson. So a lot of you might know Alan Turing. He's widely considered the father of computer science and artificial intelligence. You might also know him for breaking the Enigma code in World War II, or maybe you know him from that movie, The Imitation Game, that came out. At least that's how I first heard of him. So probably less known is his contribution to mathematical biology. So the field of morphogenesis and its name itself was really pioneered by Turing in his seminal paper, The Chemical Basis of Morphogenesis. The question that concerned Turing is, how do genes affect the structure of the eventual organism? That is, you start out as a ball of cells. How do you grow limbs, lungs, horns, stripes, or skin? How do you turn into a butterfly or a koala or anything like that? And this is a question that really still puzzles scientists to this day. It's still very, very well studied in developmental biology. But the way Turing first attempted to answer it was an investigation of chemical processes. So he developed the theory based on two pretty simple processes: chemical reactions and diffusion. So diffusion itself is a stabilizing process, of course, and in the absence of diffusion, the reactions are stable. But Turing showed that with both processes together, you could have an instability, which is now called a diffusion-driven instability. And that could eventually drive an inhomogeneity to a heterogeneous pattern of chemicals. And he called these sort of chemicals morphogens because of their ability to sort of generate shape or form. This was one of the first examples of sort of the concept of emergence, which is now a really fundamental concept in complex systems theory. You can think about emergence as sort of new, unexpected behavior that emerges from nonlinear interactions within a system. So in particular, Turing studied a system of two coupled reaction diffusion equations, one that represented an activator and the other an inhibitor. Later, Meinhardt and Greer would follow up by presenting sort of the following argument. So you can imagine that the activator promotes the inhibitor, right? Which in turn inhibits the activator. So normally you would imagine that this would lead to sort of a stable state, right? A negative feedback loop in a sense. But if we allow the inhibitor to diffuse faster than the activator, then we could break the equilibrium. And locally, this could maybe look like a circle, surrounded by a ring. So you could imagine like a lot of systems that have this sort of physiology. For example, a system that I learned about recently was the Lateral Geniculate complex. It's also known as the LGN, which is an area in the visual pathway. There's like this receptive field. There are on-center cells that react more strongly to light than their surrounding circular area. That could be something that could arise from these sort of Turing patterns. But if you're wondering about the actual validity of these models, it wasn't until about 60 years later, recently in 2014, that scientists finally confirmed that Turing's theory, and they showed that you could have all 6 different patterns that Turing predicted, as well as an unexpected 7th one. So you can see that this is still a really rich field of study that's still motivating scientists today. Now I'm going to consider another model, a really interesting model, or at least one that I found interesting, with only 3 interactions. You have diffusion, where particles sort of bounce around randomly, sort of Brownian motion-like. It's limited because there's only a small number of particles floating around, and there's aggregation, as in the particles stick together and they form aggregates. So this is a process known as diffusion limited aggregation, or DLA, as I'll henceforth refer to it. And it was first proposed by Witten and Sanders in their paper, Diffusion Limited Aggregation, a kinetic critical phenomena, to model aggregation in which diffusion dominates as the primary means of transport, as opposed to something like convection in some, Bernard flow, or active transport in some biological system. So DLA lends itself rather simply to simulation on a computer because we can embed it in a lattice rather than running the simulation on a lattice-free model. So on a lattice, we can consider some cellular automata model that would, say, start from some inertial seed in the middle of our grid, then introduce another particle somewhere outside the grid and wait for it to diffuse and stick to the original seed. If it sort of wanders too far away from the original, we kill the particle, as in we don't consider it anymore, and we introduce another particle. And if it sticks, we introduce another particle. And we do this until the aggregate reaches some predefined size. Additionally, we can vary some other properties of the particle, which we'll talk about later, and that will lead us up to some interesting characteristics. So, how do DLA clusters really arise? As you can see, the reason for the branching structure is sort of as follows. So, first, the particle, if you look at that figure on the right with the few grids, the particle is more likely to stick to the bud because from the top, it can come from 3 directions, right? Left, right, and down. Rather than one of the other particles, because that can only come from down. And secondly, the particle coming in from the outside of the cluster, as we see in this figure on the right, is more likely to be caught on one of the branches rather than near the middle of the cluster. As you might have guessed, DLA clusters are fractals, though they don't have the exact self-similarity like the Cantor set or the Koch triangle. Instead, they have a statistical self-similarity, and that's because of the stochastic nature of the process that generates them. So the branches near the boundaries can also be shown to have multifractal scaling. We won't be discussing that, but if you want, it's referenced in number 4. You can also introduce stickiness into the system, so as in, we don't always let the particle stick, but it sticks with some probability when it encounters a block near it. So, as we can see, decreasing the probability of sticking actually increases the fractal dimension of the DLA cluster. And it's not really that hard to understand why. The less sticking gives the particle more chances to move towards the middle of the cluster. You can also do DLA models in 3 dimensions on a lattice. The visualization is a little bit more difficult to get down on MATLAB, so that figure on the left is not very good. But all the code that I use to generate these DLA clusters in 2 and 3 dimensions is included in the GitHub. So github.com/arsuendra/DLA. And you can use that to generate these DLA clusters, plot them, calculate their dimensions and whatnot. So be sure to check it out if you're interested. There's also an interesting connection between DLA and the Healy-Shaw problem. That is that the idea is that DLA is sort of a stochastic version of that. And the Healy-Shaw problem, which concerns sort of flow of fluid sandwiched between the 2 pieces of glass. I didn't look too much into it, but be sure to check it out on the reference 5 if you're interested. So what can these DLA clusters be used to model? Well, generally they're used to model dendritic growth, especially in like metals with a crystalline structure, sort of electrodeposition models. However, you can also think about them in biology as sort of a rare particle and like the food of a cell. You know, the particle diffuses around. If it hits a cell, the cell eats it and it buds. So this will lend itself sort of naturally to coral-like growth or neurons neurons like dendrites, bacterial colonies, the bronchioles in the lungs. This is called branching morphogenesis, which we'll talk a little bit more about that later, and sort of the early regime of some epithelial cells, like mammalian epithelial cells. That's in reference 13. So there's a lot of other ways people study this concept of biological morphogenesis, and it's sort of been aided recently with these powerful advances, right? Like gene editing, and like in terms of CRISPR, really strong microscopes, new fluorescent biomarkers that can be used to really precisely track what happens to a cell. So we're going to talk about some of these next. Mechanical modeling is sort of one of the other two topics, I guess, or sort of the other two main ways of studying morphogenesis besides chemical methods. And you can think about it as sort of, let's say, the frills on a lizard, right, on the left there. You can have two substances on a layer, but one grows faster than the other. So this forms sort of like when that happens, you can sort of have like a buckling, and that forms these frills. And scientists can really precisely sort of model these with mathematics. You can also kind of think about plants. Plants' cell walls have a lot of pressure on them, and these can be modeled with both mechanical methods and also these chemical methods with reaction diffusion equations. People have also been studying vertex models, so instead of modeling a cell as a particle on a lattice, you model a cell as a polygon, and the lines sort of correspond to the cell walls, so you can imagine the cell sort of growing or shrinking with some rules. And there's this really interesting paper that I found that sort of combines that 3D vertex model with a Turing mechanism, and you can introduce, like, you can have all sorts of interesting things come out of that. People also study branching morphogenesis, so in the lungs and the kidneys, right? We need a really high surface area to volume ratio. So it's been studied that these structures form fractals. There's these kind of bifurcations that allow them to branch, and there's diffusion, limited growth, and whatnot. And there's this really interesting paper that I found on sort of Bayesian approaches. This one I didn't really read too much into, but it's very mathematical and it seems very interesting, so definitely check it out. So finally, these are all the references I have. What I recommend is if you have something you want to check out, just go to here, and this sort of encodes what it is on the slides before. So this is 1 to 5, 6 to 10, just pause it, 11 to 14, and there's also some other references that are not numbered in the presentation, but these are sort of what I drew inspiration from, I suppose. All right, that's all. Thank you.